2017 Biology Unit 1, Paper 1. So, let's look at this. Pistia stratiodes, right? What it is a rosette-like plant found floating in ponds and natural waterways. Which of the following properties of water assists the plant to float?
A, sir.
Speak up.
A, sir.
A. Cohesion. Alright. Which of the following combinations correctly describes the structure of molecules 1 and 2?
C, sir.
This is well, we're going backwards though, but this is a repeat. Right? So this is a constantly repeated question, right? Third, 3. 3 is a high repeat question. The answer is B. To save more time, let's just answer the questions I have repeated and then move on. Alright? 4. Which of the following combination of properties of glucose is as a result of its simple ring structure? So I don't think that 2 will be in there. I think that these 2 will be fine. 1 and 3. Do we agree?
Yes, sir.
Alright, B. For 5, what is the highest level of okay, we saw this already. Here now. Students conduct a Benedict's test or the Benedict's test on 4 different clear solutions containing various amounts or varying amounts of a reducing sugar. The results indicating the degree of color change are shown. So we have clear blue. Right? So we have clear blue there. Then we have a little yellowish here. Then we have a little orangish here. Then we have a little reddish here. So which of the following combinations show the correct sequencing of the solutions in ascending order from least amount of reducing sugar to the most amount? So it would be solution 1, solution 4, so B is the answer. Make sense? Agreed?
Wait, sir.
Alright.
Sir,
Oh, we don't agree?
wait, sir, wouldn't the brick red one be like, this is least amount to most amount, sir?
So least. So if we see a clear blue solution, right, does that mean that a reaction occurred?
No.
No. So
No, sir.
So that means that there's either no reducing sugar there, or there's such a small amount we still see a clear blue solution. So 1 has to be the least amount. Agreed?
Oh, yeah, oh yes, sir, this is right. So I was looking at the numbers at the top. My bad.
Or you're looking at the numbers. Oh, okay. So that's fine. So let's look at this. Which of the following structures present in this cell would not be seen in a bacterial cell?
D, sir.
Okay, so bacterial cells are prokaryotes, right? Before a nucleus, right? So here we have a repeated question. B is the answer. So it shows tissue. And that tissue would be what? Xylem or phloem?
Phloem.
Alright. This is repeated also. So we have to identify a eukaryotic cell. So it would have an endoplasmic reticulum, it would have mitochondria, it would have a nuclear membrane. Would it have 70S or 80S? Without 80S, right?
Yes, sir.
So B is the answer. Okay. Bobby, okay, we see that it's gonna be out of the fish. We're just gonna mark what's already repeated. When a plant cell is placed in a solution which is less, okay, we saw this. So we say it's less negative, so it's more positive, right? So we're saying, what are we saying here? So it's not more negative, but less negative.
It's in a hypertonic solution.
What does hypertonic refer to?
Sir, higher potential compared to the other one. So A.
I'm not hearing like clear, like clearly the voices of y'all. I'm not hearing like clear answers. Like this is that and this is that. Go ahead.
Yes, sir.
Because the solution has a higher water potential than the plant cell.
Yeah, so it's gonna have a higher, a more positive water potential. Let's not say higher, but more positive. So what type of solution would that be?
Hypotonic.
Hypotonic.
Hypotonic, and that would mean that water would go in, right?
Yes, sir. So what did you say not to say?
Right. Don't say higher water potential. So more positive, right? I know that you can say higher and lesser, right, and lower. I mean, but I like to see like more positive or more negative water potential because you'll make the connection in your head between the negative numbers if they bring them and positive numbers if they bring them, right? So water potential. So water potential hypertonic solutions, right? So we just need to understand those type of things, right? So we're saying that since we know the difference between water potential and and the solute concentration, right? So if you have more solute, there's a higher water potential, right? You guys,
Higher solute is more negative water potential.
more negative water potential. All right. So the more positive one means there's just more water generally. All right. So we're saying that it's gonna flow into the cell and become turgid, or flow out of the cell and become flaccid.
Into the cell, sir.
Into the cell and become turgid. All right. 12. Which of the following is not a rule of phospholipids in it? Okay. I think we saw this, right? What would be the answer?
Yes, sir.
D. Directly move. So it doesn't directly move. There are channels for that. Right? So which of the following is the best example of anabolism? So we saw all of these before. Pepsins, okay, we saw the pepsin one before as well. A would be the best answer here. Right? So we're seeing this now. With reference to the diagram, which of the following is is correct about the rate of reaction between X and Y?
D, sir.
There would be no reaction. So you don't see any complex here. So there's no enzyme substrate complex because they're not connected to each other. Alright, so we have that there. Which of the following is found in both DNA and transfer RNA? Repeated question, right?
Yes, sir. C.
Alright.
So one
17.
question about 15, sir.
Oh, what
So
about 15?
like
Go
with
ahead.
with the induced fit model, it's like when the enzyme is able to change its shape slightly to accommodate the substrate.
Mm-hmm.
What is the difference in how they look for a reaction to take place? Like how different do they have to look?
How different? Okay.
Like for example, like
go ahead.
X and Y were like combined together. It's like we said that no reaction would take place. Is it that like those squibbly like regions wouldn't accommodate it, or would it be able to like somehow expand itself for it to be able to fit?
Okay, so this is the enzyme, right? So it will change these regions here will actually change to fit, right? So it's for example like a glove, like your hand on your glove, right? The glove, right, doesn't have the shape of your hand, right? So it's closed on itself. So when you start to push your hand into the glove, it takes the shape of your hand. So that's how induced fit works. So when X starts to go into Y's active site, then it will start to mold around that complete shape of X. But when X is outside of it, then the enzyme rests in that molecular shape. Did I answer your question or not?
Oh yes, sir, you did. But in reference to like,
Okay, go ahead.
I'd said D also, but thinking about it, if aren't we talking about when X and Y like actually combine together?
Oh, okay.
Yeah, so why would interaction then take place after?
No, if they're combined together, then a reaction would probably take place.
Also, we're just taking the diagram as it is, that they're not together.
Yeah, so it doesn't show a complex.
Okay, so all right, thank you.
All right. So 17. Which of the following diagrams illustrate the semi-conservative method of replication of DNA?
A.
A. All right. Here, as this repeated, we have tyrosine and histidine. Right? So this repeated as well. Now this repeats as well. So we have chroma. Ooh. All right. So we have at 4. We have what? What is that for? Chromatin, right?
Yes, sir.
So this is the best answer. Yeah, this is the answer. I don't think there's an issue with this answer though, because it doesn't say chromosome is a chromatid or anything like that, so it's fine. Okay, so the following diagram shows the structure of a ternary molecule. Which letter A, B, or C, A, B, C, or D, indicates the position of the molecule where the amino acid would be attached? Where would it be attached?
A, sir.
A, everybody agrees?
Yeah.
Yes, sir.
Alright, would be A. 21. Which of the following best explains why DNA replication is important in maintaining genetic stability? DNA has to replicate accurately to allow offspring to inherit genes from parents. When new cells are made, each must receive 2 exact copies of the instructions to function properly, and the DNA must be replicated for cell division to produce cells that function efficiently. What is the answer?
D.
Right. So my interpretation for this, right, is that some persons would say that there are only 1 exact copy and not 2 exact copy that will go to the cells within that, right? But we're looking at, so we're looking at this, right? The purpose of DNA replication before meiosis, right, is to duplicate the amount of genes that are present, right? So it starts at 2n, then it goes to 4n, right? Then it splits to 2n, right? So after you replicate it, each cell after generally the first stage of meiosis, right, will gain 2 exact copies, as you see in 2, and then split out into n. So that's my interpretation of this question. So I think this now, the showing the 2 copies would be in relation to this, which is important. So I think D is the best answer. This is the best answer. Alright. Here now we have a repeat question. Right there, DNA synthesis fertilization, that is fine. Right, remember that fertilization is from N to 2N, so it has to be here, and that is corresponding with Y. Right, and then we have DNA synthesis, right? When DNA is synthesized through, usually replication is the name of the game, it goes from 2n to the 4n, as we saw in the question above, right, for X. Here now, which of the following is the genotype of one of the recombinants? This is a repeated question, right? But the letters have changed, or the letters changed on 2022, I believe. Right, so what would be the answer here?
A, sir.
That sounds like a question.
So the answer is A.
Alright, so it would be A. Alright, 24. Fur color. Alright, so fur color in mice is controlled by a pair of genes occupying different loci. Right? Capital W is the presence of color. Common W represents albino. Right? So we'll see epistasis there. The allele for brown fur is capital A and black fur is common A. So what is the genotype of this? So we see that small w, so it allows the presence of color. and capital A is dominant over the not submissive, recessive A here. So it's gonna be brown.
Wouldn't it be albino?
Oh, let's
Since
see.
they said common W represents albino.
Exactly, sorry. I didn't cut it properly. Albino, so it's gonna be albino. I mixed up capital W and common W, right? So what is the phenotypic ratio of the second filial generation for a cross between two first filial parents, I guess, with the genetic, with the genotype here?
See, sir?
C. All right, we didn't. Oh, wait. We did something similar to this, right? Before we did something similar to this before. So C will okay. So first of all, let's look at what's happening here. Do we all agree with C? Hello?
Yes, sir.
Yes, sir.
Yes, sir.
Alright, so we could show the working for that on paper twos. Alright, so which of the following is considered an advantage of somatic gene therapy? What is a somatic cell?
Body cell.
Okay, a general body cell, alright? So what's considered an advantage of somatic gene therapy?
B.
A.
Yes, sir.
Sir.
Oh, advantage. Oh, okay. Yeah.
Huh. Okay. Somatic
Sure.
gene therapy. Go ahead.
Well, alright. It seems like you were going to do it, but I was asking you to run through that. What is that?
Okay. So a somatic cell is a regular body cell, right? So it's not a gamete generally, right? So it's, for example, your skin cells that replicate through mitosis, right? Rather than your gametes that will replicate through meiosis, right? So we're looking at what are the advantages of gene therapy on your regular body cells, right? So we're saying that the genes and vectors prompt immune responses, not generally. Alleles are always delivered to a cell in the tissue. That I don't think has much thing to do with it. It would probably use to treat genetic disorders. Right? So you're born with a genetic disorder, right? And then after that, now you have to do somatic gene therapy, right? So general gene therapy, for example, with sickle cell, right? So 27, which of the following describes protogeny?
B.
28, in populations of Pacific salmon, okay, we saw this already, this is disruptive, right?
Yes, sir.
Alright. 29, mutations can have varying effects on an organism depending on when it occurs. The occurrence of a mutation has the greatest effect during
A.
A. Are we sure?
So either A or C.
The mutation can occur within DNA replication, right? But its effects are usually seen in translation when it translates to the protein and the protein is incorrect. Right, so it's usually mostly in translation, right? When it reads for the protein and the protein is incorrect. For example, right, mutations can occur, right? We know the deletions, the inversions, and frame shift mutations and stuff like that, right? But let's say that we're having sickle cell disorder, right? The sickle cell mutation can be there within genetics, right? Within the genetic code, right? But the major effect occurs when it translates that to a protein that causes the blood cell to form the sickle shape. So I would believe that the greatest effect will be during translation when you're seeing that the proteins are incorrect and form either disadvantageous structures, right, or structures that don't work at all, really. So we have that there. Disadvantageous structural forms. 30. Which type of isolation plays the most significant role in allopathic speciation? This repeated.
So
Sir,
Yeah, go ahead.
if 30 was St. Patrick, which one would be more significant?
If it was St. Patrick, alright. If it was St. Patrick. If it says St. Patrick, probably I'll go for temporal. Alright? Probably I'll go for temporal.
Okay, thanks.
Alright? So in plant tissue culture, small pieces of tissue can grow into whole new plants. In addition to inorganic ions and sucrose, which of the following essential growth requirements are needed to make this possible?
D, sir.
D as in dog,
Yes, sir.
auxins, vitamins, and cytokinins. So remember your plant hormones, auxins and cytokinins will be needed, right? And you're gonna need vitamins, that's what we're saying? Are we sure that we're gonna need vitamins?
I disagree.
You disagree, okay. So tell us why we disagree or why you disagree.
Because in the growth medium, we usually use the growth hormone, and you would need vitamins for the plant to grow because it's a part of the minerals, vitamin and minerals, and the C word.
And the C word. Oh boy. So gonna need the growth so you're saying that everything is needed, right?
That's what I think.
So you're saying that the vitamins is a part of the minerals. Vitamins and minerals are two different things. That's why we call them vitamins and minerals. So we're gonna have the iron. So we know we need the phosphate and the magnesium and stuff like that. We're using sucrose here as energy as an energy source, right? As well as we're gonna need auxins and cytokinins. But do we need vitamins? I'm just asking that.
Yes, sir. We don't need vitamins.
So do we need vitamins?
You can Google it. Try Googling it.
Okay, let us try to use our understanding. So usually the proliferation of like the cells, right? The growing of the cells. I think the vitamin that we normally use is like B, right? Is it that the vitamin that we use? Using B1 and B6, right? I think it's B specifically 1 and 6. Right? So we're gonna need vitamins. 32. Sexual reproduction would be most advantageous for the survival of a species if what? A new viral disease appears, environmental
A.
conditions, okay.
It's a, sir.
Okay. A horticulturalist has bred a hibiscus variety which bears violet flowers. Which type of method of propagation would be most appropriate for a rapid production of a large number of clonal plants?
Yes, sir.
A.
Is it true?
A.
Eh? Okay, somebody said B. Why B? We're looking at the most appropriate for rapid production, Fragmentation can help produce clonal plants, but it's going to be moving much slower. Tissue culture is actually moving much faster than plant fragmentation. Right? 34. Which of the following methods of asexual reproduction is incorrect? By its definition. Alright. So binary fission, splitting of two daughter. Interesting. Alright. Fragmentation, splitting of several. Alright. Budding, production of outgrowth from the. Alright. Lightweight
D.
spores.
The example is wrong.
The example is a flamboyant hibiscus. Alright. We know that they don't use spores. Right. 35 is a repeat. What's the answer? D.
D.
Beautiful. Which of the following does not promote cross-pollination? Clostrogamy.
A.
Beautiful. 37. In plants, the ovary is best described as the...
C.
C.
Alright. 38. The following are advantages of two types of pollination. Right? Which of the following are specific to cross-pollination?
Sir, C.
Okay, so we know that one is out. 2 is true. 3 is true. No, that is not true. Alright, so seeing that C. Okay. Here, no, I'm not going to even entertain this question. 40. How does a secondary oocyte differ from an ovum?
C.
It
Yes,
is
sir.
one more polar body in the secondary. Alright.
It's the
Okay, there is one more polar body in the ovum. Then okay. Do you guys have a grasp on oogenesis? We have a grasp on oogenesis, right?
Somewhat.
Somewhat. So look, I have to look at oogenesis for this now. For the what would be the primary oocyte?
Sir B.
Oh boy. I'll ask what a primary oocyte is, but okay. Let me see. So we know what polar bodies are, right? So we know that polar bodies normally form through what we call asymmetric cytokinesis, right? So we know the cytosol, right, and some of the organelles are put into a small body and released, right? The secondary, so the secondary oocyte, right, during meiosis one, right, some of the cytosol and the organelles are shipped into it, right? And then into the cell in the egg, sorry, in meiosis 2, right? For the primary oocyte, right? The primary oocyte, does the primary oocyte have a polar body?
Yes, sir.
How many?
Just one, sir.
How much would an ovum have? What is an
There's a-
ovum? Let me ask.
Sir, the female gamete.
The ovum?
That's
Nothing
complete.
else.
That's secondary oocyte after fertilization.
Foundation.
After fertilization, right? So does it still have a polar body?
No, sir.
No. So if we're saying it doesn't, what is the answer?
C.
We're saying that is the answer based off the polar bodies. So let's look at A, right? The nucleus is haploid. Never mind, let's not look at A. For B, the nucleus is diploid in the secondary oocyte and haploid in the ovum. Alright, is that true? We want to evaluate all of them, you know.
That's not true.
That's not true. Why? Why is it? Are you saying that the ovum is not haploid? The ovum is fertilized, right? So why won't it be haploid?
It's fertilized, so it has both the genetic material of the mother and the father.
Alright, so that's why it's out. So it's saying that C is the best answer here because of the position, the polar body within secondary oocyte, and then after fertilization, there's no more polar body for us to look at, right? Make sense?
Oh, so I have a question
Go
though.
ahead.
So you would consider the like it completing the meiotic division, that's still like in fertilization? Because like
No.
so how does it have one less polar body?
Oh, this has one less polar body. We're gonna have to look at oogenesis through that. We can have a look at oogenesis after, right? So let's
Oh,
just complete just 5 more and then we can look at the oogenesis. I'm gonna bring up the diagram and we'll look at it. Right? The hormones X, Y, sorry, Y, W, X, Y, and Z, I'm going blind, are, this repeated, right?
Yes, sir.
The answer is B?
Yes, sir.
Alright. So let's look at this, right? 42. A sample of a woman's this repeated, right? So we show that ovulation has occurred. Right? 43, this repeated.
42 is wrong. It's supposed to be D.
It's supposed to be D? Alright.
D, sir.
Okay. So yeah, that's what we had in other papers. My bad. Oral birth control pills will most likely, or most directly, will work most directly by.
B.
B.
So we said the mini pill do D now. Now we're talking about oral birth control pills and it's still D, right? Is that what we're saying now?
Sir, wouldn't it be the same D, sir?
Repeat.
Wouldn't it be still D?
Okay, somebody said that the mini pill that we saw in the other questions, right, looked at the thickening of the cervical mucus, right? So will it change? Will the answer change? Victoria says A. Why A? Why A? I'm waiting for somebody to come and explain to me why A. This is a more general case. Okay, go ahead, Kashim. So what Victoria is saying that for mini specifically it's D. But since we're just saying overall, we're saying it's A. We're saying generally it's A. That's what Victoria's answer is. So do we agree with her?
So I'm not sure because the oral birth control pill, isn't that something as just a mini birth control pill?
Mm-hmm. So we're asking now if the wording of the question changes the answer.
Yeah, because like I don't think there's like a big pill compared to like a little pill. I just feel like it's just an oral pill in general.
Wait, hold on. Let us answer 45. Pregnant women are advised to increase their intake of fluid. What is the answer for this? This repeated, right? B.
B, sir.
So 44 is the issue. So we need to decide that. So if it would be B. This is interesting. Oh yeah, it would be B and not A, right? Well, it would be between B and D. I'm just seeing it.
Yes, I see why it could be B, sir. But like, if we're going to give the mini pill D, sir, then what stops us from giving that or the oral birth control D also?
Yeah, that's true. If the answer for the mini pill question is D, why is this answer B? Go ahead.
So would it be because they have two? Well, it's a mini pill, so I think it only has progesterone, but oral contraceptives have both estrogen and progesterone. So that's why this makes more sense. Two different type of pills.
Okay. let us have a look at them after this. Alright? Let us have a look at them after this. So that was the 2017 table.